Hello everyone, welcome to RBR Life Science. In this video, you will learn about continuous strict method of isolation of microorganisms. The first step in this technique is flame sterilization of the wire loop. Hold the loop in the flame for a few seconds. When the wire loop is red hot, then hold the loop away from the flame and allow the loop to cool for a few moments. Now the loop is sterile and you can take out the culture sample using this sterile loop. Dip the loop in the test tube containing a liquid sample having a mixture of bacteria. A loop full of culture sample is taken on the wire loop. Now take a sterile and dry nutrient agar plate. Open the lid of agar plate for inoculation. Place the wire loop containing the sample near the edge of the petri plate in area A. Spread the culture by streaking loop in a single continuous movement to the center of the plate. When the streak reaches the center, then rotate the plate 180 degrees. Now use the same loop to streak area B. Without flaming the loop, streak the remaining portion of agar surface in area B. Hold the loop in flame to sterilize and keep the loop aside. Replace the lid immediately and invert the plate, label it and keep the plate in an inverted position for incubation in the incubator. Incubate the plate at 37 degrees Celsius for 24 to 48 hours. After incubation, remove the plate from the incubator and check for the growth of bacteria on agar plate. You can observe the isolated colonies in the center where the streak in area B was ended. So friends, thank you so much for watching this video. If you want to watch such more videos, then please like, comment, share and subscribe our YouTube channel RBR Life Science. For more detailed information, you can visit our blog rbrlifescience.com. The link to our blog website is given in the description.